Our speaker today, it's a real pleasure to introduce Sarah with an H. Peterson. I don't even know that I really have to now. There are a few people. So Sarah came from University of Western Washington. Okay, from a master. I don't know where you got your Whitman, that's right, Whitman College. So she worked with a number of people who many of us in the marine mammal diving community know quite well. At Whitman College, she worked with Chuck Drabeck, who was a guy that went with Jerry Koyman in the old days when Koyman was a graduate student. Chuck was a field assistant with Jerry, and so he wrote me this incredible letter saying this person walks on water. So how could I not believe that? And then she did her master's with Alejandro Acevedo, which many of you may remember. Alejandro was a postdoc here with Don Kroll and was the first one to write the paper about swimming with your mouth open and how the hydrodynamics of swimming as a fin whale. So again, I got these great letters. How could I not believe them? And then I met Sarah at a conference, a marine mammal conference, and she gave a really nice talk, and then she said, well, how do you do some of these analyses? I said, oh, send us your data and we'll help you. And then for months, I couldn't find anybody in the lab to help us get through the data, and then nothing happened. And so after even all that, she still applied here, and not only applied, but came here after I never followed through with helping her with some rather simple analysis. Anyway, the last thing I'll say about Sarah, because she's just been a tremendous asset to the lab, she came here and within a very short period of time showed that she really could get up to speed in no time. While I was gone last week, she had was just down, 2 weeks ago or so, on San Nicolas Island working on a California sea lion project with Paul Panganis, and many of you know Gita McDonald. They're doing a field project with California sea lions. Sarah went down as a field assistant. I got a really nice email from Paul Panganis telling me how wonderful she was in the field and that I had a really excellent student, which I already do that, but it's always nice to hear that. So Sarah's going to be giving us her proposal defense seminar.
Thank you so much for coming. I'm really excited to share the proposed research for my dissertation, which is focused on persistent organic pollutants and heavy metals in mesopelagic or deep ocean foraging seals. And I'm really interested in the influence of ecology and physiology on the contaminant assimilation, how these animals are actually getting contaminants, and then temporal tissue fluctuations, so how their extreme physiology and life history characteristics actually influence the contaminants that are in their body at different life history points. So contaminants come from many sources. They're both naturally occurring and anthropogenic. Mercury is the first of the two heavy metals that I'll be examining. It does emit naturally from volcanoes and from deep sea hydrothermal vents. But it also emits from a number of human sources, including the burning of fossil fuels, waste incineration, dentistry, all sorts of reasons that it actually gets into the environment. And this is a figure here. It's difficult to see the time scale, but it goes from 1300 to the present. And these are fur samples for polar bears. It's actually showing from these old samples the increase in mercury that's occurring as a result of human activities in the environment, a rapid increase. Lead also is naturally occurring in deposits in the environment, and it does erode naturally into the environment. However, we also are extracting these lead resources, and through lead smelting and a number of other uses of lead, gasoline and so forth, we're also increasing the amount of lead that's in the environment. The other big category of contaminants that I'm looking at are the persistent organic pollutants. These are organic compounds that are resistant to biological degradation. They don't go away very quickly, and we make them and put them into the environment. The first of these is a group of chemicals known as the PBDEs, referred to as brominated flame retardants. And they do actually find their place in a lot of your common household furniture. There's flame requirements, and every piece of upholstered furniture has actually got a lot of these in it. So we're definitely, this is a problem that is happening right now. DDT and PCBs, on the other hand, are legacy persistent organic pollutants. They haven't been legal to use in to manufacture in the United States since the mid-70s, but they've remained pervasive in the environment. They've spread around the world, and they still are being used in some places. DDT commonly as a pesticide, and PCBs used in electrical transformers. Now, both PBDEs and PCBs have a number of different chemical forms, and these are called congeners. There's 209 different combinations of ways that chlorines, in this case of a PCB, or bromines, can attach themselves, all of those little numbered positions or places that a chlorine or bromine can attach themselves to the backbone of that, what makes that chemical unique. And those different forms will come up again in my research. They move differently through animals and the environment, so that's important to know. Contaminants are a global issue. This is a figure here showing global mercury emissions. In darker blue is higher mercury emissions. You can see that it's not an even distribution of where mercury is coming from. What I'd like to draw your attention to is that this whole region up here in the north is not emitting any mercury. Yet at the same time, we can detect mercury and other environmental contaminants in polar bear tissues from the Arctic, in northern fur seals from the Pribilof Islands. So as these contaminants get into the environment and they move around through the atmosphere, they move around through the ocean currents, they're ending up in these far-ranging, far-away places, far-ranging animals in faraway places. So I'm interested in northern elephant seals that forage broadly across the North Pacific, far away from sources of human emissions, and what they may be accumulating for these contaminants. I'm also bringing in Weddell seals, which actually are from the Ross Sea, sort of the edges of this map, and trying to bring them in as a comparison because they're actually in a place that is relatively isolated from those global wind and ocean currents. So I'm trying to see how they compare to elephant seals. Now these contaminants biomagnify at food webs. So you start with the bottom of the food web, and there's a degree of contamination in the system. And as the predators eat that bottom of the food chain, those contaminants are concentrated in their tissues. And as you move up to the next food chain level, like a predator here, those contaminants become even more concentrated. So there's this magnification effect and this concentration as you move up food chains. Not only those where magnification, there's also bioaccumulation that occurs in these long-lived animals over the course of their lives. So again, these chemicals are difficult to break down in the body, and so they accumulate over time. And they can cause a number of problems. All of these, both heavy metals and persistent pollutants, have different ways in which they're affecting the body, but some, just to sort of summarize a few of the ways that they can actually affect biological organisms. Through the immune system, there have been studies showing a decrease in lymphocyte efficiency and proliferation, so the immune system's ability to fight off pathogens and really defend itself can be compromised. The endocrine system, you can have interference with hormones. They mimic hormones. These persistent organic pollutants can then upregulate or downregulate crucial physiological pathways in the body with fairly damaging consequences. And they can affect the nervous system. So mercury is a potent neurotoxin, can get into the bloodstream, readily cross through the blood-brain barrier, and interfere with the central nervous system. So it can also do things like alter the neurotransmitter receptors in the brain. So there's a number of pathways that these contaminants can affect biological organisms. So the root of my dissertation is in this simple premise that you are what you eat. So all of these contaminants are out there in the environment, and as a marine mammal is foraging out there, it's going to be picking up contaminants and representative of the place where it's eating. So if an animal is foraging in a place with more contamination, it's probably going to have more contamination in its tissues. If it's foraging higher on the food chain than its neighbor, it's going to have more contamination. So the ecological processes are going to be affecting what we see in that animal. Additionally, concentrations are based on an amount of something. So if you have an amount of contaminant in an animal, and then that animal changes its body mass by about 40%, the concentrations of that contaminant in that animal can change. And this isn't well addressed in a lot of studies because it's really difficult to follow the same animals in wild populations over time. So I'm going to be looking at both the ecology and the physiology and how they influence what we observe for these contaminants. So the players for my research are, first and foremost, the northern elephant seal here. Northern elephant seal males and females This is a figure showing where those animals broadly go in the North Pacific. So the males are in red here, foraging along the edges of the continental slope, continental shelf, and then the females actually encompass not only the open ocean, but also along the coastal areas as well. Very wide-ranging. This is the Weddell seal here, and I've been really fortunate to be able to include this in my dissertation research because of the study concurrently going on, funded, that's primarily Kim Getz's dissertation research, and they've been able to bring contaminant samples back for me so I can compare some of what I find in elephant seals with this other predator. Now what's unique about both elephant seals and Weddell seals is that many of the marine mammals out there, the seals and sea lions, spend a lot of their time foraging in this surface zone, the epipelagic zone, 0 to 200 meters in depth. But Weddell seals and elephant seals spend the majority of their time foraging in the mesopelagic zone, this 200 to 1,000 meter depth. What's really exciting about this is that there are a lot of other more cryptic species that don't come to land to breed. We don't have easy access to them. And so these two species can actually give us a lot of insight about what's happening in the environment in this far away and deep place in the ecosystem of these oceans. So the overarching questions that I'm hoping to address through my research are, first and foremost, what are the levels of persistent organic pollutants and heavy metals in elephant seals? Secondly, what ecological variables correlate with increased contaminant concentrations and blubber burdens? So again, a lot of this work is done on concentrations, but I'm actually trying to quantify an amount of contaminant in the adipose or the fat tissue, and then actually look at how that changes as well. I'm interested in how do fasting and reproductive states affect tissue concentration, so that physiological component. And then last, how do elephant seal heavy metal concentrations compare with a similarly foraging seal from a more pristine habitat? So the rest of my talk will be structured through giving an overview of the methods that are sort of applicable for all of my chapters, and then break down my 4 chapters into PCBs and females, and you'll see these little pictures will follow through as I'm talking about different components of this. Chapter 2 is persistent organic pollutants in males and female northern elephant seals. Chapter 3 is focused on mercury in males and females of both elephant seals and Weddell seals. Chapter 4 focuses on lead in both species, and then I'll briefly summarize. So what's really amazing about the system that I've been able to access by being in Dan's lab is that in studies of contaminant research, oftentimes we don't know a lot about the system. When you're studying a wild animal, they may be hard to capture more than once. You may not know how old they are. You may not know where they're foraging. But we actually can account for and examine the implications of all of these. So we have the ability to satellite track these animals and know exactly where they're going in the ocean, where their geography is, both two-dimensionally and then their spatial use of the water column. We can use stable isotopes of carbon and nitrogen to look at relative trophic position of these animals, where they're feeding in the food web. We can follow the same animals at multiple times of year and look at the dynamics of how their body fluctuations when they go from a nice fat animal here to a nice skinny animal at the end of the fast. We can look at how that affects those contaminant concentrations. And then I can look at these really interesting questions surrounding sexes. A lot of times it's difficult to study males, or people just haven't done as much study with males because they're not transferring contaminants to their offspring. But we have the ability to actually look at both females that can offload and potentially and they give away some of their contaminant concentration to their offspring, and then males which don't have that ability. And then by studying a population of animals that we know a lot about, I can actually use known age animals and look at the effects of age. So to introduce you briefly to the elephant seal life cycle, this is the year in the life of an elephant seal female, and the portions of this in tan are when the animal's on land. When the elephant seals are on land, they're completely fasting. So we have a breeding fasting period here that they're starting right now. Then they go to sea the entirety of the time they're at sea for foraging. They don't come to land at all. Then they return to Año Nuevo further molt, where they actually grow a brand new set of fur and skin, and then they go back to sea for a long foraging trip. Now, when I look at the males, what I want to draw your attention to is the fact that they have those same on land and at sea trips and time periods, but the breeding is actually much longer than for females. And this could have significant implications for what happens over the time that those animals are on land, with the amount of body mass loss that's going on and what's occurring. To also just draw your attention again to the differences between males and females in terms of their foraging behavior. So males are foraging along the continental margins here, and they're up here in the corner so you can see them a little bit clearer. They do cross the open Pacific, but they pretty much do that just to get to their endpoint, which is either out here in the Aleutians or the Gulf of Alaska. Now the females are going broadly across the Pacific out here to these patchy but rich prey areas known as the transition zone. They also will forage up along the coastal area as well, and I'm especially interested in where these females and males may be overlapping. Additionally, there are some differences in their diving behavior that may affect their exposure to contaminants. Females have these nice dives in the middle of the water column. Even the females that are diving, that are moving along the coastal margins will have a lot of, they'll dive actually in the middle of the water column. Whereas the males are actually going along and they're foraging on the benthic sediments on the bottom. So that can affect potentially what they're exposed to. Now, if we compare this briefly to Weddell seals, we have 2 fasting periods for elephant seals, one of which is associated with breeding and one of which is associated with molting. But for the Weddell seals, there's just 1 fasting period, and it's actually fairly unclear if it's a complete fast or if there is some limited foraging. It appears there might be. Kim's research will be looking a lot at this as she explores the isotopes, so that will help inform what I see. And then the rest of the time of year, they're actively foraging. So what I'm doing is sampling at the extremes of the body condition for both of these species. So more specifically, I'll take samples from a late fasting animal. We'll attach tags to that animal. It goes to sea, and then it comes back nice and fat. So I have a before and after that foraging trip sample. Then I have another set of animals that I sample at the end of the molting fast. They go to sea, and then I get them when they return at the beginning of the breeding season. So when we go out to Año Nuevo, we're looking for these flipper-tagged individuals. We know how old they are. We sedate them, glue satellite tags, time depth recorder tags, send them off to sea after taking some tissue samples. And I'll explain more as I get into the specific chapters. We additionally take length and girth measurements. We take blubber thickness, and we take mass. And what that allows us to do is actually quantify a percent of fat tissue in that animal. And this is really especially important for these persistent organic pollutants that are actually binding to fat tissues. And then we can calculate a contaminant blubber burden. So here is a little schematic of a blubber core. So you have the skin out here and the muscle. We have an inner blubber and an outer blubber. And a lot of research has shown that these are actually being used for different things in the animal. This inner blubber is metabolically active. It's what's being used to fuel the animal as it's fasting, whereas this outer blubber is thought to be more of a thermoregulatory mechanism. It's actually not really changing nearly as much. So we want to look at these separately. Then we can get a concentration or an amount of concentration of contaminant, and then we can scale that up to an actual amount of contaminant in that tissue store. So to introduce you briefly to how I'm going to be looking at some of the geography of these animals, there are these oceanographic provinces or ecoregions that are broadly defined as the California current upwelling, a very productive region, the Alaska downwelling region up here, which is another coastal region, and then these 2 open ocean regions, the Pacific subarctic gyre and the North Pacific polar front. Now there's several different ways that I'm going to be looking at geography. First, I can just break them into these coastal versus open ocean seals, and then I can also come up with some more continuous variables, the distance, the average distance from shore, the depth of the water column where they're foraging, and actually try to look at their foraging behavior in several different ways to really make sure that I'm capturing any trends that are there. So again, you can see those foraging tracks and how they would fit into the coastal versus open ocean. Additionally, I'm taking carbon and nitrogen isotopes from these animals, and carbon and nitrogen both have a heavier and a lighter isotope. And what this allows us to do is calculate a delta 13 carbon and a delta N15 values. And very sort of simply, you just look at the ratio of these two isotopes in your sample that you take from an animal, and then you compare that against the standard. And it allows you to look at those tissues in sort of isotope space. I'm going to be looking at red blood cells, which give us an approximate diet, an integrated diet over the last several months of that animal. So the way this looks in isotope space is we have carbon down here on the x-axis, we have delta N15 on the y-axis, and if we put a potential food web up here, or a food chain rather, what we have is a value down here for the primary producer. And the primary producer, this value, where it falls along this x-axis, that's dictated by the type of primary producer it is, or where it may be in the ocean. I'll explain that in a moment. And then what you can do is look at how your mid-level consumers and your predators are situated relative to the base of the food chain. So there's an approximate enrichment that happens with each trophic step, and so you can use this information to try and estimate where in the food web that this animal is foraging. And then the lab analyses themselves will take place in Belgium for the pups. In Davis, I'm running all of these on a direct mercury analyzer in collaboration with a USGS scientist up there. Lead I will run here at the UCSC Trace Clean facilities, and then stable carbon and nitrogen isotopes I'm running here at the UCSC Light Isotope Lab. Sort of the framework. And then when I get into the statistical analyses, some of which I've done, but the majority of which are ahead of me, I'll be using a combination of linear regression models and linear mixed effects models with the NLME package in R. And the reason that I'm doing this is that I do have repeated samples for individuals, and so I'll be able to account for seal as a random effect. Use AICC for model selection, and then when I figure out the appropriate ecological variables or physiological variables to include in the best model, then I'll refit that model at the end and run the appropriate post-hoc tests. So my first chapter is looking at the effects of geography, fasting states, and age on PCB concentrations and congeners. And I want to know, are coastal and open ocean foraging seals different? I also want to know if tissue concentrations and congener profiles differ between time periods, and if age has a significant correlation with the total concentrations or congener profiles. So these tissue samples were actually taken before I got to Santa Cruz in 2005 and 2007. Again, spanning the short foraging trip and the long foraging trip, the inner and outer blubber, and the serum. And these tissues are taken because these persistent organic pollutants are fat binding, and so the places to measure them in an animal are within the fat tissue, and then also in the transport system, the serum. 23 congeners, 23 different chemical structures were tested for, and I've grouped those into 4 that I used for analyses based on the number of chlorines that occur in each of those congeners. In these animals, I have satellite tracks, fasting or reproductive state, age, and the percent of fat tissue. So very briefly, when I looked at these a couple of different ways, it actually appeared that there was no detectable coastal versus open ocean signal. So in red here, we had the seals that were 11 out of the 42 seals spent a significant portion of their trip in a coastal area. The seals in purple here, although they did come out further into the open ocean, spent a significant portion of their time, the majority of their time, in the California current. And then these are the open ocean seals. They did not seem to stratify based on where they were foraging. And there's a couple reasons for this. It may be that they're far enough off the continental shelf that it's just a very diffuse and fairly even distribution of PCBs. And so I'll be looking into this further in my second chapter. When we actually look at the fluctuations between time periods, what we have here are the 4 different sampling time periods. And we have inner blubber, outer blubber, and serum. Inner blubber is here in light pink, outer blubber in purple, and serum in yellow. And we have a concentration of PCBs in parts per billion on the y-axis. And what we have are skinny animals that are at the end of their molting time period, and they're heading out to sea. And that's when we have the highest average contaminant concentrations in the inner blubber. These animals have been fasting for a long time, and additionally, they have not been transferring any of their contaminant load to offspring, because this is the non-lactation, the non-breeding fast. When those animals come back, so same animals, you can see how much lower the concentration is in their inner blubber. Their outer blubber and their serum change a little bit, but not nearly as significantly. And then when you have another set of animals going out at the end of the breeding season, again, their concentrations are higher than they were than other animals were at the start, at the arrival back from a foraging trip. And then these animals go to sea and they come back fat, and again, the mean is lower when they come back. Now, what I just want to the main take-home message from this is that these different layers of these different tissues in the animal are not behaving the same. And so this is important to understand when we're trying to figure out how at risk these animals may be. In the grand scheme of things, these concentrations are not very high for a marine mammal, but they do have them, and there are significant fluctuations over the year. When we look at the relationship with age, each of the dots up here is an individual. There is not a detectable age relationship with the total amount of PCBs in inner blubber or in outer blubber. However, when we actually break this down and we look at the chemical composition of the different structures within the blubber layers, we actually start to see in the inner blubber here a significant negative relationship with the group of chemicals that have 5 chlorines. So here we have 3 and 4 chlorines, 5 chlorines, 6 chlorines, and 7 chlorines. And we have a positive, significant positive relationship with the percent of these 7 chlorinated, this group of 7 chlorinated biphenyls. So it's kind of interesting. Something's going on here as these animals get older. Their concentrations aren't changing, but the kinetics of these chemicals, there's something going on. When we look at the outer blubber, this is a much more striking picture. We actually have, for both of the low and penta 5 CBs, we have this negative relationship, and with the chlorine, the 6 and 7 chlorines, we have a positive relationship. And what this suggests, and there's been some other studies that have looked at components of this, it suggests that there is something that's happening when these females are getting older as they go through reproduction and lactation, and they're able to transfer chemicals to their offspring, that they're more easily able to transfer these lower chlorinated structures, and they retain these higher chlorinated structures within their bodies. So, in summary, for this component, it does not appear at this time that coastal and open ocean foraging seals are different. The tissue concentrations do differ between time periods, but it's mostly in the inner blubber. And age has a significant correlation with the congener profiles, but not with the total PCB concentrations. So just looking at PCBs and seeing what I've come up with from this, it really made me want to expand this to look at other persistent organic pollutants, to include isotopes to try to get at foraging position, and to expand this to include males. So my second chapter is focused on persistent organic pollutants in male and females, and I'm trying to dig more into the details of the ecology and the physiology. So what ecological factors correlate with increased total persistent organic pollutants, several different kinds, or the blubber burdens and the actual amount of contaminant at different time periods? Does fasting condition or reproductive state affect the persistent organic pollutant concentrations? And is persistent organic pollution consumption detectable over a foraging trip? If I estimate the amount of contaminant at the beginning of a foraging trip and at the end, can I detect an actual ingestion of contaminants over the course of that trip? So these samples were taken in 2010 and 2012. The last batch of females is maybe not the appropriate term. The last females are at sea and they'll return in January, and I'll get the follow-up samples from them. We're taking inner blubber and outer blubber again, as well as serum. And then we're going to be testing for multiple types of persistent organic pollutants and chlorinated pesticides in Belgium this summer. Again, looking at 23 congeners and grouping them into families. And then I'll be accounting for fasting and reproductive state using satellite tracks and diving data from the females, mass and the percent of adipose tissue, age, carbon and nitrogen isotopes, and then comparing both males and females. So to briefly go into the messy situation that is carbon isotopes in the Pacific, we have the California current upwelling and the Alaska downwelling provinces. And in these regions, we have high delta carbon-13 values, we have low delta carbon-13 values, and then as you move out the Aleutian Island chain, there's a gradient out there as well. The big point from this is that I do realize that isotopes are quite messy, and so this is going to be something that that I'll have to really sort of massage and work with to figure out how to get the best information out of these isotopes. But I already have a lot of values, and I want to share those with you. So what this figure is showing is all of the isotopes for the animals that we've handled over the last 2 years. And along here is carbon again. So if you remember, actually, so back to here, there's this high to low gradient as you move as you move latitudinally up towards Alaska. And what we have here for males in blue is a pretty strong gradient. So that's likely driven by latitude. As you look here in the nitrogen direction on this isospace plot, you can see that females and they're just the two different foraging trips, the long and the short trip, are in the two different colors. You can see a spread from 13 to almost 17, so that's a trophic level and a half. Now, where this I think can be extremely useful is when I figure out that two animals are in the same approximate region geographically from their satellite tracks, and I can take a cut through and say, are those animals doing the same thing isotopically, or are there some differences there that may help explain differences that I see in the contaminants? And these are where the animals from last year went across the Pacific. So this is a really exciting set of data to get into. We actually had several individuals that stayed within the California current the entirety of their time in sea, which hasn't been observed all that many times throughout our previous elephant tail tracking. So it'll be interesting to see if those animals show us something different with contaminants. Now that wraps up the persistent organic pollutant portion of this talk. Now I'm going to move into the heavy metals and describe why I think that mercury is going to be an extremely interesting heavy metal to study in elephant seals. So this figure here is showing the average depth of occurrence and the log concentration of mercury for a whole bunch of fish, swimming large fish near Hawaii. And the take-home message from this figure is that these fish here are foraging at depths of 300 to 600 meters. And they have higher mercury concentrations in their bodies than these epipelagic fish from the same areas of approximately similar size. So this and several other things out there really got me thinking that there may be actually something pretty interesting happening in this zone where the elephant seals are foraging. And we don't have a system like this to be able to study what's happening in the mesopelagic, because most of the other predators in that area, we don't have the same kinds of life history information for, and we can't account for age and all the things that we can do with elephant seals. So I'm looking at mercury in both elephant seals and Weddell seals, again bringing Weddell seals in as a comparative species because they're in this relatively isolated habitat. And I want to first know, within elephant seals, are the tissue concentrations significantly correlated? So if I take samples from different tissues, can I infer a concentration in one tissue by having a different tissue? And that helps just us understand what's going on in the body. Second, when ecological variables are correlated with increased concentrations, I think you see a trend in the questions that I'm asking throughout these chapters. How does the fasting state and the body condition affect tissue concentrations? And do elephant seals have higher concentrations than Weddell seals? So again, these samples were taken from 2010 to 2012, and I'm finishing up sampling this next year. We looked at blood in both elephant seals and Weddell seals, and then muscle and fur in just elephant seals. I tested for total mercury, and what I didn't mention earlier is that there are multiple forms of mercury in the environment. Mercury that is in its inorganic form and floating around in the atmosphere or being emitted from hydrothermal vents is not toxic. It's not a bioavailable form. But when it gets converted to a form called methyl mercury, that's when it actually can start interfering with the body's processes. In all of these tissues, blood, muscle, and fur, the majority of the mercury that's in that tissue is the bioavailable kind. It's the toxic kind. And that's why I'm able to just test for the total mercury in that tissue. And then for these animals, I also have, again, the fasting, reproductive state, satellite tracks and diving data for females, mass and body condition for females, age, carbon and nitrogen isotope, sex, and percent of red blood cells. So mercury is actually a protein binding contaminant, and so that's why I'm measuring it in these tissues. And the amount of red blood cells that you have in your blood, the proportion of red blood cells, is going to influence these contaminants. So that's something else we can account for. Now again, you've seen this figure before, but what I'm also going to be doing, instead of just sampling around a foraging trip, is actually take one animal, or several animals, do this for the same thing, take a late fasting sample, let that animal go to sea, take a sample when it comes back, let it fast, take another sample from that same animal, and let it go back to sea, and then take a sample at the very end. And this is an animal that is satellite tracked and has all the equipment on it. So what this looks like when you spread it out is that you have 4 samples over 2 foraging trip periods and a fasting period. And I'm doing this for a subset of animals, so we can really look at the dynamics of their body condition fluctuation across this whole time. And then I'm actually doing this for another subset of animals during the breeding fast, so I'm going to understand the dynamics there as well. Now, some preliminary data from the seals that have come back already. What I'll be doing is doing a plot like this and getting really into the nitty-gritty of these analyses, taking the muscle concentration here and the blood concentration, and really figuring out if there's a strong relationship between those 2 tissues. So this is actually preliminary analysis shows this is a pretty strong relationship, but you can see it's fairly messy. There's a lot of noise around that. When I actually break it down by the specific time periods that I'm sampling, you can see that there's a much stronger and cleaner relationship between blood and muscle at these time periods when an animal has just come back from foraging. When those animals are at the end of their fasting period, that's when this relationship starts to break down a little bit. So this information will really allow us to understand the dynamics of how the contaminants are moving throughout the body and the implications for contaminant monitoring at different times of the year. So these are, again, more preliminary data. I'm going to walk you through those 3 different tissues. So these are late molting, early breeding, late breeding, and early molting. So we have a skinny seal here. We have higher concentrations for males than females. Then the seals come back, and we have lower concentrations in the blood for both the males and the females. So there's something going on here. Seals are, again, at the end of their breeding fast, and the contaminant concentrations appear more elevated than they were during that the early portion of breeding. And then animals come back from a foraging trip and early molt, and again, from this time period to this time period, those concentrations have changed. So there is a significant influence, it appears, that occurs over the course of the year with these fasting conditions. Now, when we look at muscle, it seems like there's going to be a different story here. You actually have, these are the same time periods, you have not very much change occurring in the females, but you do have a significant increase here. Again, the statistics have not been what appears to be a fairly important change during the late breeding. And so it'll be really exciting to get in and see if this is driven by, again, that long period of time that these animals are, that those males are spending on land at that time period. And then when we get into fur, we have the scale here is 0 to 80 parts per million, and so I didn't, I'll show you where these fall in the grand scheme of things in a moment. But this is relatively high, and these concentrations here were fairly striking. and I'm excited to talk to people about more of this later, but the females are relatively consistent when it comes to their fur concentrations over the year, and there's something interesting happening here with the male fur concentrations. If you remember, the molting period is when the animals are growing their fur, and so it may be something to do with the actual seals seals having mercury go into their fur during that time period, and at the time period that we sample those animals may influence what we actually observe as far as the concentration in that fur. So my collaborator has done a lot of work with feathers in birds, and they have a nonlinear deposition of mercury in their tissue. So as they start to grow their feathers, more mercury is getting put into their feathers early on in that period. And then, so that may actually be something that's occurring with these elephant seals. So what these three figures have basically shown is the very early stages of what I'm going to be getting into as far as understanding how the fluctuating body conditions of these animals influences their contaminant concentrations. And I'm also going to be turning this on its head a little bit when I actually look at these animals across the fasting period as well. So we've now flipped the positions. So this is a fat animal here that is going to, the average, lose about 35 to 42% of its mass over the course of its time on land, and it becomes quite skinny. And these are the concentrations that I'm observing. Now, I will say this is not the same individual here. So what I'm going to be doing in the future is actually taking the same individuals, and so we can very specifically link the dynamics across time within an individual animal. And then again, the same thing over this molting fast, and they lose approximately 25% of their body mass, and see if how that corresponds to their contaminant concentration changes. When I compared these blood mercury concentrations to elephant seals, from elephant seals to Weddell seals, it appeared actually as if Weddell seals were doing something similar with their blood, where in that late fasting time period where there may be some opportunistic foraging, those concentrations may be higher than they are when they're just arriving back to the ice to start giving birth to their pups. But the scale is quite different. So when we actually shape up elephant seals with Weddell seals, we see that the blood mercury parts per million here is really low, significantly lower than the elephant seals, almost an order of magnitude lower. And so it really does appear that that system, a similarly foraging predator, similar depths in the water column, actually really isn't it is not accumulating similar levels of mercury. It may be that Antarctica really is quite isolated. Now, when I put these into the overall scheme, what you have here are a lot of animals. So each of these bars and standard deviations is a different study that I found from the literature. My values for elephant seals for males fall here in this red box, and for females here. And these are fur concentrations from published studies, and these are most of the published studies for seals and sea lions around the world. And you can see that the elephant seal tissue samples that we're observing here are on the higher end of that. What's around those are harbor seals. So I would have expected, actually going into this study, that I was going to find a lot lower concentrations in elephant seals, an animal that's foraging thousands of kilometers in the middle of the ocean, versus harbor seals from Germany, from California, from Denmark, that are foraging right along the shoreline in places where they're fairly close to human populations. But it appears that elephant seals may be right there with them. Now, when we look at where this places them in terms of potential toxicity thresholds, some animals were above the threshold for observed neurological effects in mink. The sort of threshold that's been put forward for neurological effects in terrestrial wildlife. And a lot of these animals, a lot of these species in general, but definitely almost all of our elephant seals, fell above this subclinical neurological effect in polar bears. Now, I'm not saying all of these animals are having neurological problems, but it's interesting to think that that in polar bears, what they were actually able to show was that there were changes in the brain chemistry, the decrease in neurotransmitters that occurred that they would not have been able to detect by just looking at a polar bear. And these animals that we're studying are going through these long fasting periods, and if they're elevating their mercury at the end of a fasting period, right before they head out to sea, and this is a neurotoxin that can decrease their ability to forage, there is a potential for an impact. And it will be interesting to sort of play around with what that would mean for these animals as I go through this. And then US EPA guidelines for mercury in human hair is actually down at 1 part per million, so most of these animals are above that. When I look at it in muscle, because the muscle is actually sort of an integrated value of what's happening to that animal at that moment in time, you can see that they're even on the higher end of the scale. Now, I've been able to bring in marine foraging birds, cetaceans into this as well, and even bluefin tuna. So all the tuna recommendations, don't eat more tuna than this because you're going to accumulate mercury. They're way down here. Elephant seal mercury in their muscle is way up here. So that's pretty striking. So it appears that blood and muscle may be correlated. I'll get into the specifics of all of those other tissue correlations as I go. I'm currently analyzing the movements in diving in relation to mercury concentrations, and I'm currently analyzing the fasting state and body condition in relation to mercury concentrations. And then I'll be finishing taking up the rest of the samples this year and looking across the fasting state as well, and really getting down to the individual level for each of these animals. What does the individual animal's change in body mass mean for its change in contaminants? And elephant seals do have significantly higher blood mercury concentrations than Weddell seals. Now, the last portion of research that I'm doing is the least developed, but I think actually could be, has really interesting implications. So I'm looking at lead, and lead actually has 4 stable isotopes. So you can look at the ratio of, say, in this figure here, 207 to 208, and 208 to 206, and you can do that for all of the combinations of lead isotopes. And what that allows you to do is actually identify the likely source of lead to that system. And this has been done successfully by a few people here at UC Santa Cruz, actually, and it will be really great to work with them and try to make sense of what's happening in elephant seals. So this has been done to show sea otter bones from pre-industrial and post-industrial sea otters from the Pacific actually having a difference in terms of the lead in their bodies. And this is the figure shown here, actually. So what this is showing us is that on this axis is 1 lead isotope ratio, and here's a second. And the natural sources of lead eroding from the sediments where those animals are overlaps exclusively with the pre-industrial sea otters. And the aerosols over here and the emissions from the nearby, or the potential nearby sources of lead to the environment overlap with the post-industrial sea otters. So there has been a difference in what those sea otters are being exposed to. Albatross in the Northwest Hawaiian Islands was looked at by Myra Finkelstein, who's a former EEB graduate, and she was able to show that lead-based paint from a decommissioned military base was actually getting into albatross using this technique. And then lead poisoning of California condors is currently being examined by Myra up in Don Smith's lab, and she's using this to trace the source of trace lead bullets into California condors. And so what I'm excited to look at is, first, is it detectable? Is lead detectable? And I think it will be, but in low concentrations. To look at then, what are the potential sources of anthropogenic lead to the North Pacific and the Ross Sea? And this will be a big search through all of the literature for the different ratios of emissions that are coming out of Asia, North America, and then the naturally occurring isotopes in the environment. And then to look at whether lead concentrations in elephant seals are actually different from just the concentrations themselves, different from samples collected 20 years ago. there was a master's thesis by Kim Clucker that actually looked at this, and so I'll be able to compare the samples that she had with what we observed. And if I look at basically the animals over the last 2 years where they've gone to sea, and these are just females, I have samples from males too, I'll be able to pick individuals that spent a significant proportion of their time in different regions. So these animals were really spending their foraging time here. These animals spent their time here. These animals were in this region, coastal animals, and then a seal up here in the Aleutians. I'll be able to pick animals that are non-overlapping and actually see if their lead isotope ratios structure them geographically, and then actually see where the potential sources for lead are from in those particular systems. So in summary, this is an incredible system. These animals can serve as sentinels of the places where they're foraging. I'm really excited to be able to look at the ecological factors and how they actually are affecting what we see in these animals. Again, the North Pacific is this really large place. These animals do an amazing job of covering it, and they can tell us a lot about what's going on there. And then I can actually look at really, really fine-scale effects of changing body condition on contaminants, and this has really significant implications for contaminant monitoring in the contaminant field, because we have so much data on the life history of these animals that can really inform our analyses. So with that, that's what I'm doing. That's what I'm planning to do. This research has all been made possible by everyone who's helped me in the field over the last couple of years and will continue to help in the future. And I just want to thank everyone in the lab for helping me make this work. All of the people involved in Año Nuevo. This is a massive effort to do what we do with elephant seals. Everyone at Long Marine Lab, my comms committee, all the important friends and family in my life, my cohort, and thank you to my proposal committee, Pete and Dan, and then 2 of my members who can't be here. Kathy's in Belgium, and Josh is at USGS up in Davis. And lots of funding sources for the archive data and for the currently collected data. And with that, thank you guys so much for listening.
You showed that there were, with the PCBs, the different congeners, there's that relationship with age.
Yeah.
Is there different physiological consequences to having the more chlorine, like the different congeners? Are there different effects of those?
There are some, and that's something that I have yet to get into. There are some that are more toxic than others, as far as where the ones in these seals are falling out. The concentrations are still relatively low, so it's likely that there may not be an effect that we're seeing, but yeah, it's definitely something to consider. There are differences, for sure. Or,
does data exist, or are you getting data, or samples, I should say, of species lower in the food webs, so that you can build a coherent food web for any or all of the contaminants that you're using?
No, I'm not. Actually, I talked to my collaborator about that, and mercury is really size specific, so that would be pretty much its own, more than a dissertation, to actually sort of quantify. If you have smaller fish and larger fish of the same species, you would have to run a tremendous number of samples. So it would be really cool to do, but I'm not currently doing that.
There was that graph you're looking at with the mercury concentrations of fish in Hawaii, and I was kind of curious. It was kind of blurry. I couldn't read all the names of the fish. Were they kind of all around the same trophic level? I recognized some tuna, and so I was kind of curious if the reason that the mercury concentration was higher is because you were looking at fish that were foraging deeper, might be bioaccumulating more, versus foraging shallower.
So I'm gonna have to go back and dig out the specifics of the paper, but no, it actually really was a depth thing. So they did account for the fact that it could be a different trophic level, could be a different size, size and trophic level would affect that. And so even accounting for that, there still was an increase in. And what I didn't show is actually that there was this really cool study that happened a few years ago where they took archived feathers from a bird that feeds on deep ocean animals that come up to the surface, and a bird species that feeds on shallow water, and the magnitude of the increase in mercury concentrations was just like off the charts, but higher for the mesopelagic feeding bird. So it seems like there's really something interesting happening in the mesopelagic right now.
Really good job. That was awesome. But I was wondering, have you guys taken milk samples to see, to kind of confirm if the lower, I think it's chlorine, the ones with the lower chlorine rings are actually being offloaded, if that's how it's getting out of the system? Or someone else has looked at that?
Someone else has actually, yeah. So my collaborator Kathy actually does a lot of lactation physiology with persistent organic pollutants. I'm not sure if she's specific. I don't think she's published on that, but she's looking at milk. And then there are a number of studies that have actually shown that the proportion of congeners in pups, because they've taken actually pup blubber in serum as well, and what's showing up in the pups is different than what's in the moms, and they do have more of the low chlorinated. And it appears that the mammary gland is actually acting somewhat as a filter, and it's kind of a cool. It's just kind of crazy what's going on, and I don't understand the kinetics of it really, but there's something there.
I should know this, but you're looking at organics, mercury, and lead, and I can't remember whether you've got samples from individual animals for all three of those. So are those, you do?
Yeah.
And do you know whether are they all independently accumulated within an animal?
Yeah, so the persistent organic pollutants are going to be accumulating in the fat mostly, and fat is not really a reservoir. There's a little bit of mercury that accumulates in the fat, but not much. So they're accumulating in different body compartments. And then lead actually is a biological analog to calcium, so it actually accumulates in the bone. I'm not sampling bone. So it's only short-term exposure that I'm looking at in the blood. So there's a possibility they could be interfering, but I don't think so.
I was actually going in a different direction, which is, one of the things that you're thinking about is the relationship between, say, body condition and these things. And you can turn that around too, right? And say, what is the relationship between these things and body condition?
Yeah.
And whether that's a synergistic effect of
on the body.
Yeah.
Definitely. I mean, I think the other component is that if the period of your life as an animal that you have the highest concentrations it's when you're weaker for other reasons too, that you're at a negative disadvantage. I mean, you're at this disadvantage. Now you're going back to sea, and you're trying to recoup all of what you lost. If you have a neurotoxin in your body, and you have other things that may impair your ability to fight off pathogens, even if they're small, they may synergistically interact, and definitely ways that we may not be able to actually clinically observe. Yeah.
Well, here's a naive question from somebody who doesn't know anything about the animal. I'm really intrigued by the fact that they're putting all this mercury in their hair, and I wonder, is that thought to be some kind of an adaptive, like, offloading mechanism, or is that just kind of a passive, it just ends up there because that's where a lot of is?
So it's definitely working I'm not sure how it sort of works in favor or not for the animal, but in terms of a lifetime accumulation, it is a way that they can actually offload males and females without passing to an offspring. And it happens in birds, and it happens in animals that grow fur. So these animals happen to just pulse it all at once, and they grow their fur in a short period of time. So mercury is going out of their blood into their fur at one time during the year. And humans or other animals that are continuously growing are continuously locking up whatever's in our bodies into our hair as well. So we have a contaminant record throughout our hair. Birds do the same kind of thing that these guys do, which is when they're growing their feathers during their annual molt, they pulse it in. And so it may be sort of an added benefit of that molt is that they do get rid of some of that contaminant load, although it kind of getting pulled. There may be something else though going on in the body that because it's kind of getting channeled into their fur, it's going through their blood. So there may be an added risk during that time period for the animal.
Yeah, sorry, it's not really a question for you, but now I don't know to add more work, but I was just curious when you were talking about the graph where you're showing the risk to mercury and everything like that. So I was thinking, well, a lot of times people usually suggest for mercury that the pups are really the most at risk because they're developing, and that's probably the mercury having the most negative effects on the animal. So I was interested to know if you guys have taken samples from pups or looking at whether or not, I know it's been suggested that sometimes like the
first pop? Does it look the most?
Yeah.
Has the greatest chance for the highest accumulation because it's like mom's first chance to offload and stuff.
Yeah.
So I was just curious if you guys had taken samples at all or anything like that.
Yeah, so actually this coming field season, we're planning on taking fur from the pup, which will be cool because the fur will have been grown while the pup was in utero, and that's actually thought to be the time period that the majority of mercury is transferred. It's actually not as much through milk, especially for elephant seals since the milk is so high in fat that the transfer occurs while the pup is in the womb. So I want to look at fur mercury as a way to look at that. It would be an amazing study to look at the firstborns, but I'm unwilling to. I mean, we haven't done that, and I'm unwilling to do it because we're dragging. Then we'd be working with first-time moms. So it's kind of you get into the moral dilemma, and that's not something I wanted to do. Although scientifically, it's really interesting. No.
Just kind of as a curiosity, you talk about how there's that impact on neurotransmitters. Is it a dopamine-based pathway? Do you know?
There's some dopamine, and then with the GABA
Okay.
as well. So yeah, it's a couple different.
There are some refreshments if anybody wants to come.